This video will discuss enzyme inhibition, including competitive, non-competitive, uncompetitive, and allosteric enzymes. This video is presented by Medicos Online. Enzymes are biocatalysts that speed up biochemical reactions without being consumed. They achieve this by reducing the activation energy. Enzymes must be tightly regulated through enzyme inhibition to prevent product levels from becoming undesired. An enzyme binds to a substrate at the active site to speed up a chemical reaction, forming products. This visual shows the enzyme binding to the substrate and releasing the products. Enzyme inhibitors are molecules that bind to enzymes and reduce their activity. Inhibitor binding can be reversible or irreversible. Irreversible inhibitors chemically change the enzyme, making it permanently inactive by modifying key amino acid residues. Reversible inhibitors bind non-covalently, and the inhibition can be reversed when the inhibitor is removed. Most biological inhibitions are reversible and regulate metabolism. Note that activators also bind to enzymes, but they increase enzymatic activity. There are three different types of reversible inhibition: competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. This visual illustrates the three types of reversible enzyme inhibition. Competitive inhibitors bind to the active site, non-competitive inhibitors bind to the allosteric site, and uncompetitive inhibitors bind to the ES complex. Here is another visual comparing competitive, non-competitive, and uncompetitive inhibitors binding to the enzyme. In competitive inhibition, the inhibitor competes for and binds to the active site, preventing the substrate from binding. This forms an enzyme inhibitor complex that does not produce any product. Structural analogs of the substrate act as competitive inhibitors. Examples of competitive inhibitors include malonate for succinate dehydrogenase and allopurinol for xanthine oxidase. In competitive inhibition, the maximum reaction velocity, Vmax, is unaffected, but the Michaelis constant, Km, increases. Increasing the substrate concentration can overcome this type of inhibition. This illustration shows competitive inhibition, where the inhibitor binds to the active site of the enzyme, forming an enzyme inhibitor complex, which results in no reaction. In non-competitive inhibition, the inhibitor binds to a site other than the active site. The inhibitor may bind to a free enzyme, forming an EI complex, or to the ES complex, forming an ESI complex. Binding changes the structure and shape of the enzyme, affecting the reaction rate. Vmax is lowered, but Km remains unchanged. Non-competitive inhibitors, also known as allosteric inhibitors, bind to the allosteric site, causing the enzyme to change shape. Examples include some anti-cancer drugs that inhibit DNA synthesis and cyanide poisoning, which irreversibly inhibits an enzyme in cellular respiration. In uncompetitive inhibition, the inhibitor binds to a site other than the active site, but can only bind to the enzyme-substrate complex. This inhibition works best when the substrate concentration is high. Both Vmax and Km for the enzyme are lowered. This diagram illustrates uncompetitive inhibition. The inhibitor only binds to the enzyme substrate complex, forming an ESI complex. Examples of uncompetitive inhibitors include drugs used to treat poisoning by methanol or ethylene glycol. Tetramethylene sulfoxide and 3-butylthioline-1-oxide are uncompetitive inhibitors of liver alcohol dehydrogenase. Lineweaver-Burk plots are linear representations of enzyme kinetics. This visual shows the Lineweaver-Burk plots for competitive, non-competitive, and uncompetitive inhibition. In competitive inhibition, the slope changes, but the y-intercept remains the same. In uncompetitive inhibition, the slope remains the same, but the y-intercept changes. In non-competitive inhibition, both the slope and y-intercept change. Enzyme inhibitors are used to treat various human disease states. Examples include GABA transaminase inhibitors for epilepsy, MAO inhibitors for antidepressant effects, and ACE inhibitors as antihypertensives. Allosteric enzymes are reversibly inhibited or activated by metabolites that are not their substrate or product. These enzymes have an additional binding site, other than the active site. Binding of inhibitors or activators at the allosteric site causes conformational changes in the enzyme molecule. This can decrease or increase its catalytic activity. Allosteric enzymes are usually composed of multiple binding sites and often show a sigmoidal graph of initial rate versus substrate concentration. Therefore, they do not obey Michaelis-Menten kinetics. Their kinetic properties are explained by a conformational change between a low-activity, tense or T state, and a high-activity, relaxed or R state. Aspartate transcarbamylase, or ATCase, is a typical allosteric enzyme. It catalyzes the first step in the biosynthesis of pyrimidines. ATCase is activated by ATP and inhibited by CTP. The enzyme contains a regulatory subunit for effector binding and a catalytic subunit with the active site.